this grant is endowed. Dr. Insel received his MD degree from the University of Michigan and post-MD training in internal medicine at Boston City Hospital and Harvard Medical Service, NIH, and UCSF in clinical pharmacology. He joined UCSD in 1978 as the first MD in the Division of Pharmacology in the Department of Medicine, which later became the Pharmacology Department. Dr. Insel is currently Emeritus Distinguished Professor of Pharmacology and Medicine. He didn't want me to mention this, but he has received many awards. I'm not going to go through them all. Is a fellow of several scientific societies, has been editor of many journals, currently of the Annual Review of Pharmacology and Toxicology. His own research has focused on G protein coupled receptors, and he has published greater than 300 original articles, 190 invited publications. But relevant to this grand rounds and this lectureship, Dr. Insel was the director since 1989 and in recent years co-director of our medical scientist MD PhD training program, or better known as MSTP program. So the UCSD MSTP began in 1974 after receiving a training grant from NIH. And can I call you the father of the MST program? Would that be okay? All right. Well, that's kind of what it seems. So the grant has been renewed for the past 50 plus years, most recently from 2025 to 2030, which in the current environment is no mean accomplishment. The UCSD MSTP has been a launching pad for MD PhD trainees who have undertaken careers as physician scientists in academia and industry, and many MSTP trainees are UCSD faculty members, including in the Department of Medicine. Dr. Insel and his wife, Barbara, established this lectureship in 2023 to highlight physician scientists, especially UCSD MSTP alumni. And this is obviously particularly important in our current environment. Previous speakers were Dr. Will Go, Nicole Kufal, and Amir Zaranpar. And today's speaker, Dr. Gerald Joyce, who Dr. Insel will introduce, is a prime example of the highly successful careers of graduates of this program. So please join me in welcoming Dr. Insel, who introduced Dr. Joyce, and also thank him for establishing this and for celebrating physician scientists at UCSD.
I think I'll use this microphone. You were on this one, right? Yeah. Oh, thank you, Zia, for those kind words. And thanks for what's a large turnout in the audience. And I presume there's several people on Zoom. It's really my pleasure to introduce Gerald Joyce, who I think we can easily say is one of our most successful and highly regarded graduates of our MD PhD program. Jerry originally attended undergraduate at the University of Chicago, and he joined the MSTP, if I'm not mistaken, in 1982. Is that correct, Jerry?
Well, 78.
In 78, sorry, yeah. 78. He graduated in 1984, so he did it in 6 years, ladies and gentlemen, when that was actually possible. For those of you who are up, there are some MD PhD students in the audience, so you should, and maybe online. So, but Jerry's had a very illustrious career, both as a medical student and PhD student here, but then he decided to do first an internship and got licensed to be a doctor here, a physician in California. He did his internship. He'll tell you more, I think, about that. And then he went to the Salk Institute to do a postdoc, if you will. He stayed there for several years, after which he took a position at the Scripps Institute, where he spent almost 30 years at the Scripps Research Institute, during which time he did some very outstanding work that led to his election into a number of honorary societies, the National Academy of Science, the National Academy of Medicine, and the American Academy of Arts and Sciences. And he then had a sort of a slight turn. He decided to go into industry for a short time, which is what a lot of MD PhD students are thinking about doing, I think, these days. And he was at the, I'm going to let him tell you the details of this, but what we call GNF, working for Novartis here in San Diego, running their facility, the that was called GNF. After which he then went back, but this time to the Salk Institute, where he's been now for approximately, if I got it right, how many years, Jared?
8.
Let's see, I think it's since 2017. And for the last 4 years, he's been the CEO and president of the Salk Institute. And he continues to do some outstanding research, which I think he'll tell you, because his work has both had basic science interests, but also has potential clinical importance. And we're just really glad to have you come over, Jerry. For those of you, some of you may know we were going to have him last year, but he got an honorary award from in Ireland, so he had to cancel. And we're really happy to have you come here, Jerry. Thank you.
Thanks, Paul. Thank you, sir. It's an honor to lecture in the Insel lectureship. I mean, it's also kind of homecoming. You know, I did grow up here. The first time I ever encountered Paul, I was probably I used to sit in the 3rd or 4th row here. This was OPP, Organ Physiology Pharmacology. It was his first year on the faculty at Salk. And it was an intense course, but like compared to John West, he's such a nice guy. You know, if people know John West was the course director, but it was team taught. And, you know, you and Joan Heller Brown, it's like, oh, some of the faculty are actually nice. John West had a rule that he would fail 10% of the class. And so you had to do remedial over the summer to make it up and take an extra exam, including an oral exam with him. You know, it was pretty rough stuff. Everyone knew it was a 10% failure rate. So I wasn't one of those, but it was still intense. Anyway, so that's how I first encountered Paul. And of course, we've interacted quite a bit, including at GNF over the years. You know, always greatly admired his work on G proteins, GPCRs, receptor signaling. And then the great director of the MSTP program, you know, Eric Warnbrock, Paul Insel, and now it takes 2, right? You have co-directors now. As I was walking to the auditorium, I went past the old MSTP office. It's in the same place. Yeah, but it's locked down. I couldn't get in. That's like, you know, there's all these signs and beer. Anyway, it is a real pleasure to be back at Salk. As you can tell from the introduction from Paul, I'm a product of the Mesa, of the Torrey Pines Mesa, or a vagabond of the Mesa, you know, depending. I mean, so I was, of course, trained here. I was trained at Salk, many years at Scripps, Novartis, GNF, General Atomics. I still have an office on the General Atomics campus, strategy, and I bounce all over the Mesa. So I thought what I would do today is, you know, kind of hop around the Mesa and tell you some stories of what's been going on around here, especially in my life. And so it's all about life and scientific discovery on the Mesa. So I can't get CME credit for my own talk. I put in more than an hour putting this together, but I guess I can't. Okay, so is everyone done with that one? Okay, good. Yeah, because I do need my credits. Okay, let's see. Should advance? Am I doing it right? Is there a reason this isn't? Am I doing? All the lights are on. Was I too far away? Oh, okay. Just hit the center button. Okay, well, all the lights are just for decoration? Okay. Okay, this is what the Mesa, the west part of the Mesa, the bluff top, looked like in the 40s. This is Camp Callan. So I think people know UCSD back in the day was Camp Matthews. It was a Marine base. This is Camp Callan. It's an Army artillery training base on the bluff on the Torrey Pines Mesa. That's a postcard. That's not an actual photograph. But you can look on the web and see all kinds of photographs. It was quite a big operation. You know, there was a worry that the Japanese were going to hit the coast in Southern California. So their guns lined up, but also as a training base. After the war, it became a professional car race circuit. And that lasted for about 10 years. Here in the number 2 Ferrari Monza, that's the great Phil Hill. You know, Carroll Shelby won a race there. I mean, it was really big time. And after the war, car races were a big deal, but not enough of a big deal, I guess, to justify the cost of running the raceway. And then, as you know, and through modern times, golf, right? Then golf became the big thing in the late 50s into the 60s, and the magnificent Torrey Pines golf course, municipal golf course, home of 2 US Opens so far, sits there. Now, just on the other side of the glider port sits this magnificent structure. This is my current home, where I did my PhD training and my postdoc training, the Salk Institute. It's truly an inspiring, gorgeous place to be. I don't know if you've all had a chance to be there. When I came and interviewed for medical school, there wasn't much traffic on Torrey Pines Road, and I was staying at the little hotel across La Jolla Village. And I'm like, where's the ocean? So I just started walking that way, and everything was wide open then. There were no gates on the Salk campus, and I just wandered into that space. I'm like, oh my God, what is this? So this is, I'll talk more about it, but this is a National Historic Landmark, considered one of the, most seminal pieces of architecture in the country. So, so that's where I get to work now. It's a lot of fun. Every day, Corey knows, every day I'm out there walking that courtyard just for the inspiration of it, also to get to my lab, which is on the other side. So, so I'm going to talk a bit about Jonas Salk, right? So this is Medical Grand Rounds. We should all know about Jonas Salk, but maybe there's some things you don't know. So I want to tell you a bit about Jonas Salk. He was born in 1914, lower middle class family. He went to public high school, went to NYU School of Medicine, did postgraduate training at Michigan, working on a grant from the US Army to try to develop a flu vaccine, because influenza was really a threat to the troops. This was, World War II. After the war, he took a position as director of the Virus Research Laboratory at the University of Pittsburgh. And this is what he's most known for. I guess the two things he's most known for. One is polio, and the other is the Salk Institute. So with funding from what was then called the Foundation for Infantile Paralysis, founded by Franklin Delano Roosevelt, who himself had suffered from polio, continued to suffer, the sequelae of it, to try to develop a polio vaccine. So that foundation is now called the March of Dimes. So they funded Jonas Salk's work at Pittsburgh to develop a polio vaccine. Now, I don't know if anyone here, I never saw a live infection of polio. I mean, there's still polio in the world, but has anyone here actually seen, or diagnosed a case of polio? It could be coming, given certain policies. But I think anyone who's trained, has seen someone who's had polio and has the sequelae of polio, right? Has had the muscle weakness and deformities and so on. But unless you lived at the time, it's hard to realize how terrifying this disease was. So after the war, the late 40s, 50s, in the U.S., thousands, were being killed per year from polio. So there was the death rate, but there also was the fact that the survivors, many of them then had, deformities and muscle weakness for the rest of their lives. And a lot of why the disease was so terrifying was it mostly struck children. And it mostly struck them in the summer when they were out of school. And that's because it's fecal oral transmission, and kids aren't as good on their hygiene. And so when it's summertime and they're out there playing and they're in the swimming pools and all that, there would be transmission. And parents were incredibly freaked out about this. The kids were too, because they saw some of their neighbors come down with polio. The paralysis could just progress to whatever point it got to and then hopefully resolves. But if it progressed to the point that your intercostal muscles were compromised, the so-called famous iron lungs, where kids were put in these, and it was mostly kids, were put in these, these pneumatic chambers, negative pressure chambers, where a bellows at the foot end is basically, creating negative pressure to expand your chest wall to help the poor kids breathe. Some kids were in those for days or weeks, some for months, some never get out, some died in the iron lungs, and some got out, but then had sequelae from the disease. It was really a terrifying thing for the country, for the world. So that was Jonas Salk's program, to develop a vaccine against polio, against the three different strains of polio. And his vaccine is a so-called killed vaccine. So viruses, to me, are not alive because they depend on a living host to cell to propagate. But killed means that they've been inactivated, and he used formaldehyde inactivation, which a little bit of science, alkylate N7 of purines, and also irreversibly cross-linked the proteins. So they still are antigenics, they still have their shape, but they lose their function because they've been chemically cross-linked irreversibly. So he grew up large batches of polio virus in culture and then inactivated them. And of course, there was a lot of purification and all that. That was his vaccine. The Sabin vaccine is so-called live vaccine. It's transmissible, but it's a non-virulent strain. So you can get it by taking it yourself, the Sabin vaccine, or by having fecal oral contact with your neighbor. So anyway, that was the Salk vaccine. Now, In 1953, they ran a safety trial, came through that okay, not a definitive efficacy trial. In 50, ran this massive million-kid trial, and everyone wanted to get in this trial. Can you imagine that today? You know, parents clamoring to get their kids in. You were a polio pioneer? Whoa, did you get the vaccine or the placebo?
Placebo had to get a shot, second shot.
Oh, you knew that you got the real thing because they told you didn't have to get this. Oh, after the trial. Okay, very good. So it you know, kind of looks like a young you, doesn't it? Yeah, yeah. They're all grinning, you know. But I would like to think that if we had a similar emergency today, we'd see pictures like that, you know, maybe with modern clothing and iPhones hanging out. But you know, the trial was successful. The pivotal trial was successful. Last year, just about this time last year, we celebrated at Salk the 70th anniversary of the declaration of the safety and efficacy of the polio vaccine. Within a few days, it was regulatory approval, and within a week, it was being given to hundreds of thousands of kids across the country. So it was just ready to go. So, and then the cases just plummeted. Polio still exists out in the world in two ways. There's a little bit of it in the wild in parts of Afghanistan, Pakistan. There's also vaccine-derived polio because the live vaccine can mutate back to being virulent. And so that's actually the bigger problem right now. So the Salk vaccine is back in use now because, of course, it's a kill vaccine to try to do ring vaccinations around the outbreaks, either the natural outbreaks or the vaccine-derived outbreaks. But look, it's a triumph of medicine. And you know, national hero, right? Like John Glenn, Charles Lindbergh, you know, cover of Time magazine, ticker tape parades, you know, people lining up to get their vaccine, just like we did at the beginning of COVID when we first got the vaccines back in the day. So anyway, that was a really important time. He didn't try to patent the vaccine. He was afraid that if it was patented, it wouldn't get to the third world. So he refused to have it patented. You know, famously said, "You can't patent the sun." You know, so it's a little overdramatic. But and as I said, it's almost eradicated, but it's not gone yet for the reasons I said. And then the other great thing Jonas Salk did was he created the Salk Institute. So the nation was so grateful, and the March of by then called the March of Dimes Foundation, that they built an institute from him. You know, that I showed you the picture of, and he commissioned Louis Kahn, a renowned architect of the brutalist style. This is, by architects, you know, aficionados, considered to be Kahn's greatest work. He's done a number of remarkable buildings, but the Salk Institute is considered to be his pinnacle. Another one of these Jonas Salk kind of lines, you know, we want an institute worthy of a visit by Picasso. It is in its own style. And so in 1960, it was opened up. It's on land that was owned by the city of San Diego, and there was a referendum, which like 80% yes approval from the citizens of San Diego to give the land to Jonas Salk to build the Salk Institute. So we're grateful to the city as well for the beautiful property we're on right over the bluff. These are the original faculty, what were called fellows at the beginning, now called professors. There's Jonas Salk, Francis Crick, and over at this end, that's Leslie Orgel. That was my PhD supervisor, Leslie Orgel at the Salk Institute. He was one of the original Salk fellows. So we've had 6 faculty who've gone on to either before or during got the Nobel Prize. We hope Tony Hunter could be number 7, but don't want to jinx it by even saying that. But 6 so far. You know, it's a very renowned place, and it's a small place, small but mighty. There's only 50 faculty there, about 700 researchers. And what we do, you know, as Paul alluded to, we do more of the basic research there. We like to say foundational or fundamental rather than basic, because it's not simple, okay? But it's very preclinical. But we are concerned with disease. We just aren't doing clinical research, which is absolutely essential to you know, that everyone does their part in the pipeline. But Salk plays at the sort of front end of the pipeline by doing foundational research in these areas, including plant biology, by the way, for agricultural need. You know, and we have our areas of focus. We don't do vaccine research. The Salk Institute never did vaccine research. Jonas didn't want to like replicate a vaccine lab. He wanted to go after what were then the biggest and still are the biggest challenges, metabolic diseases, cancer, neurodegenerative disorders, and that's what we focus on at the Salk Institute today. So that's a bit of what we're up to. Now we're going to go up the bluff a little bit This is GNF. So this was GNF under construction starting in the year 2000. GNF is so it's just off John J. Hopkins. GNF, I should say, was, because about a year and a half after I left, GNF got absorbed into Novartis Corporate Research. And then Novartis Corporate Research got absorbed into Novartis Corporate R&D. And that kept happening. But for a good 20 some odd years, 22 years, GNF existed. And I had the privilege of being the Institute Director of GNF for 7 years, from 2014 to 2021. And it was a separate corporation, but owned by Novartis. And we at GNF worked on preclinical drug discovery, so not basic research. In fact, we weren't able to do basic research. We had to do drug discovery projects that we could hand off to Novartis that they could take to the clinic and take to patients. So about 550 employees. I would get a budget at the beginning of every year, kind of a blank check. We decided for ourselves how to spend it. We had a board of trustees, though, that we had to answer to. So we got about $150, $160 million a year to do preclinical drug discovery. And our areas of focus were cancer, including immuno-onc. These were days when Novartis Corporate Research didn't really believe in immuno-onc, but we did at GNF. They eventually saw the light on that. Regenerative medicine, autoimmune, these were kind of the key areas. And GNF was very engineering driven. I had 40 engineers that would build technologies to do drug screening, high throughput drug discovery, phenotypic screens. But I had 70 medicinal chemists to actually, make the molecules, and about 280 biologists. So it was quite a fun operation to run, and it was all about getting first in class. I used to say GNF stands for Get Novartis First in Class, because that was our goal, was to hand those things off to Novartis. Just before I arrived, Zycadia, an ALK inhibitor, was approved by FDA. That was the first drug originated at GNF that made it all the way to FDA approval. I was there to actually participate in the celebration, but that happened just before I arrived. But while I was there, we got Odomzo approved. That's a sonic hedgehog pathway inhibitor. Braftovi, BRAF inhibitor, came out of GNF, now combined with a MEK inhibitor, Mektovi, as the Braftovi-Mektovi twins, marketed by Pfizer, actually, now. But that came from GNF. And then finally, Siponimod, Mesa, the only approved drug for secondary progressive multiple sclerosis, came out of GNF. So in the end, that's what it was about. I didn't do anything. Okay, good. Okay, because I'm just about to get to a little more edgy part of the talk. Because in addition to all the fun, it really is fun and challenging of doing drug discovery, it is part of Mighty Novartis, So, Mighty Novartis is part of this massive industry called Big Pharma. So I just want to, especially for if there's medical students here or trainees, this is what this world actually is like, all right? It's a massive enterprise. The global pharmaceutical market, $1.6 trillion. I'm looking at you because I think you guys are you house staff? Oh, you're MD PhDs. Oh, but you must be in a clinical year. You're doing, anatomy lab. Thank God they still do anatomy lab. Sadly, they don't do dog lab anymore. That was one of the best parts of OPP was dog lab, because you really learn physiology based on how many ampules you're busting out. So anyway, okay, 1.6 trillion a year. And I think this is something, when you hear the word big pharma, the reason is the top 10 pharma companies each bring in between $50 and $60 billion a year in pharmaceutical sales. And I'm not talking about total sales. So the J&J number doesn't include baby powder and stuff. This is actual drug sales between $50 and $60 billion per year each. So more than a third of all that market is held by the top 10 companies, okay? These are numbers from 2025 year end. And you've heard the word blockbuster. So a blockbuster means a drug that does over $1 billion of sales in a year. But of course, what Big Pharma is looking for are super blockbusters. So a super blockbuster, and here's the top 10, do more than $10 billion in a year. Okay, any guesses what the #1 selling medicine was last year in sales? 32 point something billion dollars last year. Anybody? Good guess, but not correct. Also good guess, but also not correct. No, Humira, year after year, Humira was top until it went off patent. right? So Keytruda, the PD-1 checkpoint inhibitor, anti-PD-1 antibody, 32.3, I think, billion dollars last year. By the way, Keytruda goes off patent at the end of 2027. Yeah, so there's some concern at Merck, so that's Keytruda. Okay, yeah, the next ones are Ozempic and Mounjaro, right? The Lilly and the Novo Nordisk drugs, right? And and this is the first time they get above the $20 billion mark each. And here's the rest of the top 10. And I think Zepbound is Mounjaro, same molecule. And Wegovy is Ozempic, same molecule, just different disease indication, different marketing scheme. Shouldn't use the word scheme, but and something about the top 10, I think that's worth noting. 8 of the top 10 are biologics. Okay, so either an antibody or a peptide in the case of the GLP-1 receptor agonists. There's only 2 in the top 10 that are small molecules. Biktarvy is a 3-drug combo for HIV, anti-HIV suppression, and Eliquis is an anticoagulant. Those 2 are small molecules. Now, there's big news today. What's the name of the drug? Orforglipron was FDA approved today. So this is an oral GLP-1 receptor agonist that comes out of Lilly and was approved today. And you may have heard that recently the Novo Nordisk drug was approved in oral form, but that's still the peptide, and it's not the way to go. It's to try to get the peptide absorbed and work. It's a kludge. But the orforglipron is a small molecule, and the daily dosing is sub-milligram. It's just a gorgeous molecule. So watch, maybe not next year, but certainly by the year after. And they'll start shipping it next week. I mean, they know this is going to be a super blockbuster. So it'll be on the chart next year. Of course, to develop these drugs, you got to spend a lot of money, right? And this is, of course, what I also saw being at Novartis. So this is the R&D spend of the top 10 companies in billions, and they're all spending between $10 and $15 billion a year on drug discovery. That's about 20% of revenue. That's a big commitment. And about 1/3 of that goes to research, getting things to the clinic and early stage trials, and 2/3 goes to development, taking them through pivotal trials, manufacturing and all that. Okay, that's a big commitment of money. And you put the commitment of money dividing into the approvals, it costs, amortized over the spend, $2.2 billion per FDA approval. Okay, so this is really big business. And at GNF, we were just a small part of this. GNF was just 10% of Novartis Research, and Novartis Research was just 5% of the number of employees across Novartis. So the scale is just unimaginable. Okay, I'm going to be a little tart here, but here's something I really hate about pharma. Drug advertising, okay? Because just in the US, drug advertising last year was $15 billion. If it costs $2.2 billion to make a drug, I could get 6 or 7 drugs if I didn't do all these stupid ads, right? And the stupidest ones are the ones that are on television, right? $6 billion a year last year in TV ad spend. It's like almost 10% of all the commercials you see is this stuff, right? And like Skyrizi's #1. Why is AbbVie spending so much on Skyrizi? Because Humira went off patent, but they own this too. So they can't make as much money on Humira. It's fallen off the super blockbuster zone, but they're doing it with Skyrizi. So, et cetera, et cetera. So that drives me nuts. There's only 2 countries in the world where you can do this, direct advertisement to consumer. The United States, and for whatever reason, New Zealand. It surprises me because, anyway, and I can also tell you, and I'm not speaking for anyone in particular at Novartis, pharma hates this too, because it's an arms race. They have to do the advertising to get their market share. But they'd rather spend the money on research to make more drugs. But it's an arms race. And so I hope, and there's always, forces trying to push back on this. Why do we allow this? As doctors, those ads are awful. And then all the fast talk at the end and talk to your doctor. It should start with talk to your doctor, Anyway. Okay, rant over. Why does it cost $2.2 billion to make drugs? Well, about the clinical trial phases, I'm sure, right? There's 3 phase 1, phase 2, phase 3 from FDA filing of an investigational new drug to filing a new drug application to try to get approval. There's the 3 phases of clinical trials, and things go wrong in each phase. Now, phase 1, that's the safety study part, and that's pretty good success rate. 70% of clinical trials pass phase 1. That's because the preclinical models are pretty darn good. The animal models of disease and of potential off-target effects are pretty good. So 70% make it through safety. Then you get to phase 2, though, and that's when it gets really tough. Does it really work? And human physiology is different than any animal model. Even if you do things in non-human primates, once you get into the human and all their countercurrent illnesses and what other drugs, it starts getting really complicated. Only 30% of phase 2 trials are successful. And then you get to phase 3, which is the pivotal trial. Does it really beat the standard of care? And now the numbers in the trial are much larger, so adverse reactions that are rare pop up. So those are the 2 main reasons there's a 50% drop off in phase 3. So you put those all together, that's a 10% success rate of anything that enters clinical trials. And it costs millions to do a phase 1, tens of millions to do a phase 2, hundreds of millions to do a phase 3. So it's big bucks that are going here. But that's just the clinical part. At places like GNF and in all the big pharmas and in all the biotechs, you got to get to clinical trials. And so there are these steps that you have to go through that aren't foundational research, aren't basic science, that are preclinical drug discovery that take you from launch to IND. And they go by different names, these phases at different pharmas. But at Novartis, we use the terms exploratory, discovery, optimization, and preclinical. They're synonyms, but these are the 4 phases. Exploratory means we already know the drug target and we already know the modality of what we're trying to make, whether it's an antibody or a peptide or a small molecule. We already know what we're going after. We know how we're going after it. We just need to get our act together and get the reagents, develop the assays, and be prepared. So that's exploratory phase. So you're already doing drug discovery at this point. In the actual discovery phase is the high throughput screens, the antibody campaigns, with biologics or with cell-based things, you might be trying a bunch of different constructs to try to find hits and to try to advance those hits to what might be a lead toward a clinical candidate. In optimization, that's when the medicinal chemists do their magic and iron out all the liabilities in the molecules and really work on potency and ADME properties. And of course, looking for safety risks. There's always safety flags that have to be ironed out by the medicinal chemists or by protein engineers if it's a biologic. And then the definitive GLP toxicology studies in 2 different animal models to really be ready for the safety phase 1 and make the batch, right? The good manufacturing practices manufacturing. So those all have to happen first, and those have a 15% progression rate. So only 1.5% of all formal drug discovery projects make it. We had 30 some odd always going at one time at GNF, and we'd pull them off the hot list once they started to not progress. Otherwise, they'd progress. And fortunately, while I was there, we put 14 compounds into the clinic. What I can't do is tell you the real drug discovery stories out of Novartis because of proprietary stuff. But I can tell you a bit about one of my favorite topics, which is RNA. I'm an RNA biochemist. And when I got to GNF, we developed a strategic initiative to try to drug RNA with small molecules. We thought there are so many potential targets out there that aren't proteins, that are RNA molecules. Let's go after them because we know that functional RNAs can adopt a well-defined shape, right? And we know that those well-defined shapes of RNA can bind small molecules. This is an aptamer here binding a particular ligand, but we know that can happen. And when a structured RNA binds a small molecule, its structure changes, and therefore its function changes. So it's just like a protein, right? You want to inhibit a kinase, then you find a small molecule that jams into a binding site, either the ATP site or an allosteric site of the kinase, and causes a structural change, either a blocking of a site or a structural change, and you got an inhibitor. So we thought we could do this with RNA. And so I'm going to give you an example, but not one of the real drug discovery projects. And I don't know what's happened to these projects because I'm on the other side of the fence now. But even if I knew what was going on, I wouldn't be able to tell you. So, because it's how I signed my retirement agreement from Novartis. I was technically retired because after 7 years, I was done. I saw the writing on the wall that GNF was going to be absorbed into Novartis. But also later that year, I was turning 65, which in Switzerland, even though I was in California, hey, that's basically the end of the show. So, but we did a project that we could tell the world about using the same techniques that we use for the real targets. And the target we used in this demo project is not a clinically relevant drug target, potential drug target. It's this aptamer. So this is the theophylline aptamer, developed, back in the 80s, sorry, in the early 90s. There was an NMR structure published in 97. And it's an in vitro evolved, in vitro selected RNA that was selected for its ability to bind theophylline. It's a 33mer, and it binds theophylline with sub-micromolar affinity. And it's very selective in what it binds. So caffeine, which is just N7-methyl theophylline, that's the difference between your tea and your coffee, 10,000 times less binding or less affinity in binding caffeine than theophylline. So it's a nice model of a molecule of an RNA molecule that has a structure and binds a ligand tightly and specifically. Okay, so what we want to do is beat the theophylline aptamer to its own ligand, discover hits, ultimately leads, ultimately therapeutic compounds, except no one really wants to target this thing for therapy, that do a better job than their own ligand of getting in there and binding to the theophylline aptamer. So the way we did it was we took the theophylline aptamer, we put a fluorescent tag on it, and we took a competitor invader strand and put a quencher on it, right? And then we mixed those two strands together in the presence of different compounds, small molecule compounds. And we're looking for a compound that binds to the aptamer and tightens its structure to make it harder for the invading strand to invade. So if a small molecule binds anywhere, but say in the theophylline pocket, it'll be harder for the strand to invade, and there'll be more fluorescence. So it's a fluorescence assay for that interaction, okay? Not all of our RNA-targeted drug discovery things use this kind of assay. In fact, many of them use things that are more phenotypic, more cell-based. But this is a thing you can do where you're just going after the RNA with small molecules. And it's pharma, okay? So we went big. We used ultra-high throughput screening, 1536 well plate format, and we threw 1.1 million compounds at this assay. That was about a third of the total number of compounds we had at GNF. We had 3.2 million compounds. We threw up 1.1 million compounds at 50 micromolar concentration, and we tuned the assay, so we got about a 1% hit rate, so 11,000 hits. But a lot of those are pseudo hits, right? So then what you do is a secondary screen. We did 4 secondary screens at 2 different lower concentrations, either 5 or 30 micromolar. We repeated the original assay I showed you, but at these 2 lower concentrations. We did an assay where we added spermidine and tRNAs to diminish the risk of non-specific interactions, because the tRNA is kind of a soak. And the spermidine is, basically throwing a bunch of amines, so it's blanketing the phosphates of the RNA. We reversed the polarity of the assay, switched where the fluor and the quencher were, so that now you're turning off the fluorescence, so there wasn't a bias in the fluorescence itself. And then a completely different kind of assay called a fluorescence polarization assay. We ran all 10,700 hits through these secondary screens, and 153 survived. So that's 0.1% of all the hits. Sorry, 0.1% of the entire library, and we're down to 153. And 40 of them, and I'm not going to show you the biophysical data, but we did BioCore, on rates, off rates. 40 of them show an IC50 dose response with higher affinity than theophylline. Okay, so those are confirmed hits. Now, often what one does when one's doing drug discovery is, and here's the ones I can show you. So this isn't all of the 143. These are the non-proprietary ones among the 143. But you can see they're all different things. That's theophylline. So theophylline was one of the compounds in our deck. So it did come out, obviously. But among this zoo are things that bound more tightly than theophylline. So theophylline under these conditions, submicromolar. We call these TALs, theophylline aptamer ligand. They had different number names inside Novartis, but we're not allowed to show their names outside. So they're called TAL 1 through 4. And that's 340 times better KD for TAL 1 compared to theophylline, 2, 3, 4. Now, the next step you want to do is go to MedChem and now start making these compounds even better and worrying about all their, pharmacologic properties, their PK properties, and so on. So structure is one way to do that. So there was no crystal structure of the theophylline aptamer at that time, but our biophysics team was able to capture at roughly 2 angstrom resolution the theophylline aptamer in either the absence or presence of theophylline. And you can see in the presence of theophylline, that particular C residue, C27, gets kicked out, and the entire structure collapses around the theophylline. okay? And that's kind of how our assay was set up, because remember, we're looking to prevent strand invasion, and in this tightened down structure, strand invasion is more difficult. And of course, we validated the assay first with theophylline, methyls, all different methylxanthines, et cetera, et cetera. Okay, so here's what theophylline actually does. It sits in this wonderful little pocket when C27 is kicked out with a floor and a ceiling of the nucleic acid bases. So the ceiling is 3 different bases and the floor is 3 different bases, and it nestles in there. And at its own level, it makes 3 hydrogen bonds to other nucleotides. So you can see why it's a pretty good fit. You know, remember though, the way the aptamer was raised, you already had the molecule and then you selected an RNA to bind it. Now we're playing it the other way. We have the RNA and we're trying to discover through screening a molecule that binds to it. But that's how theophylline does it. Here's Tal1. It picks up a 4th and 5th hydrogen bond. It sits in that nest with the floor and the ceiling of other bases, but it picks up a 4th and 5th hydrogen bond. That's its secret, which we now see thanks to structure. And there's Tal2, 3, and 4, which have 3, or in the case of Tal4, only 1 hydrogen bond, and do it different ways, picking up more hydrophobic interactions and so on. So that was very instructive to us. This is how you do drug discovery if only this was a real drug target. But if we got to this point, what would we do next if we really want to take this to a clinical candidate and get to that IND step? We'd want to learn from the whole screen, not just take the one best compound and try to tweak it. So we'd want to amalgamate the medicinal chemists. We want to amalgamate all that we learn. So we want to hold on to that nice hydrogen bonding network, but then Tal2 only has 3 hydrogen bonds, but picks up, you know, nice interaction from the solvent-exposed tail. So we'd want to combine both of those. Oh yeah, and then we'd have to do all the other stuff, right? Because this is going to be a pill. So good oral bioavailability, good target engagement, good PK properties, safety, 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 right? That's where most of the compounds die, is they pick up a safety flag you can't get rid of. So if only this was a drug target. Okay, so it takes a lot of people to do drug discovery. It is, you know, this isn't rocket science. It's way more complicated than rocket science. Rocket science is much more deterministic than drug discovery. That's why the overall success rate is 1.5%. So it takes a big team to do drug discovery. And these are the people that worked on the theophylline, you know, demo project. I mean, they were working on actual drug discovery projects too, but you know, it takes a mix of these different skills. You got to have the biologists to develop the assays and the people that know how to run the assays, the medicinal chemists, and then in this case, structure was very informative. Okay, now we're going to go back across the street to the Salk Institute. So after really exciting time and a wonderful team, as you saw at GNF, the former GNF, back to the Salk Institute, just want to do basic science again, basic research. And as I said, I love RNA, so back to RNA, but something very fundamental that I've always been interested in, starting with my thesis research. How did life begin? Because as Francis Crick and Leslie Orgel said in the late 1960s, we think life began with RNA and later evolved DNA as a genetic material and proteins as the chief functional molecule. Francis Crick in 1968 said, you know, maybe the first enzyme was an RNA that was a replicator of RNA molecules so that it could evolve those molecules, like we do with directed evolution, but it did it itself. Now, that was before ribozymes were discovered. This was said in 1968. Crick also said, you know, probably the first ribosome was an RNA enzyme. You know, pretty good, huh? You know, tRNA kind of looks like nature trying to do what it does with proteins, except with RNA. Because remember, these are early days of molecular biology. So that's what I work on in my lab, is to try to make an RNA enzyme that has RNA replicase properties and can be alive, right? So not a small ambition, but we're working on it. Okay, so RNA catalyzed replication of RNA. You start with a parent strand of RNA and then you make progeny strands, right? Of course, it's Watson-Crick. So the template makes a complement and then the complement makes more template. I'm just showing it simply here. And you know, if it's going to do evolution, it'll pick up a mutation. And if it's a good mutation, it wins. If it's a bad mutation, it dies out over the course of evolution. Now, these aren't just letters of RNA, they're molecules. They're what Crick talked about, the first enzyme of life. They're replicator molecules that do the replication of the RNA. That's what we want to make in the lab, is one of those guys, okay? One of those things. So on the chemical level, a template of RNA, a primer to be extended of RNA, the building blocks of RNA, the so-called NTPs, and then the enzyme is going to come along just like a polymerized protein and zip, zip, zip it up and extend the primer and make the complement. And then use that complement as a template to make more of the original template and do that again and again. Okay, as a starting point, my friend and colleague Jack Shostak and his former graduate student Dave Bartel developed this RNA ligase ribozyme using test tube evolution technology that he and I and Larry Gold developed in the late 80s, early 90s. And this ligase does that same chemistry. It's like it's adding an NTP to the end of a primer, except it's just doing a single join. It's a ligase, all right? But then we took this ligase, and over the past 10, 11 years now, we've been evolving it using directed evolution technology to be a better and better RNA-dependent RNA polymerase. And it got bigger, it got better. It can take a primer on a separate primer binding to a separate template, extend it by adding NTPs. More recently, its structure kind of rearranged over the course of test tube evolution, and it's gotten really good at being an RNA-dependent RNA polymerase. It can actually make its own ancestor, the ligase ribozyme. So if we take a template that encodes the ligase ribozyme and a primer and give it our polymerase, evolved polymerase ribozyme, it'll extend that primer on that template to make the ligase that is its own ancestor. Obviously, what we got to get to is where the polymerase can make itself. Then it really is what Francis Crick was talking about. Then it would be self-evolving, it'd be alive. And unlike a virus, it wouldn't need like the help of some other living thing. It would actually be alive itself in a technical molecular sense. So it can make that, and okay, only one gel. Okay, I'm not going to do no Westerns, just one gel. So here's the primer labeled. Okay, back on. All right, I'll talk. We're probably almost done. Where are we on time? I forgot where we used to look at the clock all the time not when you were lecturing, but oh yeah, it's over there, right? Okay, all right, I'm just gonna wrap up here. So you can see the primer adding nucleotide after nucleotide, and it gets to 93 nucleotides long, and it's made the ligase, and then that ligase is a ligase. But we want it to be able to make itself. Can't do that yet, but what it can do is it can not just make other functional RNAs, it can evolve other functional RNAs. And that is what Crick was talking about, although not yet evolving itself. So what if the template encodes what's called the hammerhead ribozyme? I'm going to shut this off so it doesn't come on and give us feedback.
There's a whole set of batteries right here. Charged. Just take that.
Okay, back on. All right, sorry folks. All right, so the polymerase ribozyme is going to copy a template to make a hammerhead ribozyme, a smaller ribozyme, but now we want to evolve it. So we don't know if it's a good hammerhead or not. It could have picked up a bad mutation, or it could have picked up a good mutation. So we're going to now interrogate the population of hammerheads that the ribozyme synthesize. Now what the hammerhead does, it's an RNA cleaving RNA. It cleaves RNA in a site-specific, in a sequence-specific manner. So we're going to attach a little RNA substrate to the front end of the hammerhead, actually by putting it at the front end of the primer. And if it's a functional hammerhead, it'll make that cut and lop off the little RNA substrate. And then that's a basis for selection because a molecule, I'm leaving some steps out here obviously, but there's a biotin sitting there, so it's sitting on a surface, and if it makes that cut, it falls off the surface, and then we can suck it up. So only if you're a functional hammerhead do you make it to the pipette and get sucked up. And then the functional hammerheads get used as templates to make more of the original template. But through this process, mutations occur because the polymerase ribozyme makes mutations. You're going to see that as I end the talk. And if those are bad mutations, we'll lose hammerhead activity, or the hammerhead activity will go down, and those molecules will drop out of the population. But if they're good mutations, they'll survive and maybe even outcompete the others, all right? So we did 8 rounds of RNA catalyzed test tube evolution of functional RNA of hammerheads. And these are violin plots just showing you through deep sequencing the distribution of mutations. You can see the population is walking out into mutation space, right? And the population as a whole is getting better and better at getting themselves replicated and retaining function. Of course, and we published this a couple of years ago in PNAS. I'm not going to show you all the details, but of course we cloned out the individual molecules and ran them to ground, like what makes this one win? How efficiently is it copied? How efficiently is its reverse complement copied? How good a hammerhead is it? How well does it fold? What's its propensity to be air-ridden when it gets copied? All those things are part of fitness. But I thought I would just end by showing you what evolution looks like, okay? It's all molecules in clear colorless solutions of 20 to 50 microliters. So I'm not going to actually literally show you like, see? But I'm going to show you the result of the bioinformatic analysis of what evolution looks like. So I got to explain these plots. So that white dot, that's the starting wild type hammerhead ribozyme, which comes out of nature, okay? It exists in many different organisms in nature and in different forms, but it's sort of one of the canonical versions of the hammerhead that everyone references to. Okay, so that white dot is the starter, and you're going to see over time the population spread across this fitness landscape. So that contour map is the fitness landscape of hammerheadness. All right, so the higher elevation, so you can see the contours. The most inner contours closest to the white dot are the highest functioning hammerheads. And as you go further out, you lose hammerheadness, you lose catalytic activity until eventually you're just dead. Okay, all right, so deep sequencing then informs this. So after the first round, lots of mutations, okay, including things that are just lost to the solar wind. They're not hammerheads, they're not going to leave progeny, they're terrible. They're progeny now, but in the next round, these are all a goner. But the more you're towards, the high parts of the topo map, the more progeny you're going to leave in the next round. Now, the color coding refers to cluster analysis. So that's a red cluster, and the red dots are high copy number sequences within the cluster. All right, so a lot of detail, but I'm not going to just play the movie as it goes through its rounds of evolution, but I wanted to set it up. So the contour map is function. Out here is dead. Close, higher elevation is good hammerheadness, and the color tells you until it goes white. That means like blown out, bunch of singletons, right? But the red cluster and the peak sequences within a cluster is one early answer to the fitness challenge of being replicated and being a hammerhead. Okay, I'm just going to let it play. So this is what evolution looks like. Oh, sorry, I'm blocking. Darwin is so cool, So we didn't do anything except just like squirt, right? The molecules did all this. The ribozyme did the replicating. The hammerhead had to be a hammerhead, and look at the things it discovered. There's like this whole other ledge out here at high, topo elevation, and there's a bunch of sequences that founder sequence found it, and then it made more variants around it, and et cetera, et cetera. So in the paper, of course, we talk about each of these clusters and why each one rose at a particular time and why some fell off and so on. But you can see it's hanging on to information and it's evolving better answers. Now, in general, the answer is they get replicated better. They're not significantly better at actually doing the RNA cleavage. That's a really easy reaction that the wild type does really well, but the wild type doesn't get replicated very well. So most of what they evolved was being better replicants, Anyway, so that's what evolution looks like. I hope I gave you a sense of life and science on the bluff, starting from Camp Callan and artillery all the way to Darwinian evolution. These are the folks that did the work in my lab that I talked about at the end. I showed you the whole Novartis team. And NASA and a couple of foundations have funded this work over the years. So that's what I got for you today. Paul, again, honored to be speaking at the Insel lecture and kind of weird to be back in this room. Thank you all. There's still a lot of food, yeah.
A little time for questions. There are questions from the audience. Katie?
Anything's for him
You're gonna have
except
to repeat.
politics. Nothing about RFK Jr., please. Okay.
Can you, let's see. Here, let me give you a mic.
Those evolutionary experiments are quite cool. But did you see selective sweeps like you would with a virus that all of a sudden becomes super fit in that particular environment and then sweeps out all the rest?
Yes, yes. So what we didn't do was change the reaction conditions. So those were the same. So the sweeps weren't driven by, we have in not the hammered evolution, in directed evolution, done things like change the temperature, change the magnesium concentration, and they adapt. Here the conditions were the same, but it's more the stochastic effect of mutations being born, right? And then having the chance to sweep across the population. And and it's in the paper, but I don't want to go into too much detail. The higher order variants, the ones that have more mutations, of course, didn't come up at the beginning. So the first things to compete with the wild type were the single or double error mutants, because those are easy to access. But by the end, we were out 7 errors. And the ones that were the best were the ones that swept later. So that's more the stochastic effect of finding and then piling on mutation on top of mutation. And there's epistatic effects, as you might imagine, and there's contrary effects, that this mutation prevents that other good mutation from arising. So there has to be a solution then that tips the balance the other way. So that's the kind of stuff we see. Yep. And in biological evolution, same deal, just on writ large. So maybe. Okay.
So my understanding is that part of why you've been doing this, to bring back to where you started,
Yeah.
is to try to develop new potential diagnostics and therapeutics.
Yes.
Say something about that in terms of
Yeah.
where this could be.
Yeah. And so in my own lab, not. Okay. So, I got a lot to do being the president of SOC. So in my lab, we just focus on, the evolution itself and the origin of life issue. But aptamers, which I touched on, which are obtained by directed evolution, and ribozymes are used for diagnostics and have gone into the clinic. My lab, when I was at Scripps, developed the first DNA enzyme. So instead of making enzymes out of RNA, which nature has done, we made them out of DNA so that they are more stable, cheaper, and all that. And those molecules are still in clinical trials. So so they cleave the mRNA of a protein that you're trying to diminish. And so there's a GATA3 trial in Europe, and so I think the aptamers have more purchase right now. Purchase meaning, I guess, literally, because of the VC money. So there are aptamers under development for various clinical applications. Yeah, so that's where this goes for medical application.
One more time,
Yeah.
question from somebody from.
Oh, that came from online, that question?
Yeah,
Oh, okay.
no, this was.
Oh, that was you. That was you. Okay.
Charlie Goldberg wrote, looking to the future, what might be the role of AI
Oh
in
gosh.
assisting, complementing the drug discovery process?
Yes. Well.
It's a very timely.
Yeah, it depends on how far in the future we're looking. So right now, personal opinion, the contribution of AI to actual drug discovery is modest at best. You see it in things like AlphaFold, where you can at least get an idea of structure, even if you haven't made the structure, and AlphaFold, subsequent variants and related. David Baker's work are now saying, yeah, but we can even do virtual docking of virtual ligands. But if you talk to people who do real blood and guts drug discovery, AI is not there yet. It might help you, collate your business systems and I know, especially in biotechs, I don't know about Novartis, they use AI to write their INDs because there's so much stuff that has to go in those. So it sucks all their data in and it drafts the IND for the FDA, So, but the part that's going to be the last to go, where, John Henry's still going to do, is not going to give up to the steam drill, is the medicinal chemistries part is so hard. They're artists, they can look at a binding pocket, or even if you don't have a structure, imagine a binding pocket, look at a suite of molecules that bind there, either competitively because it's the same pocket, or non-competitively because it's a different place. And they can do this thing where they can imagine where in the molecule to put an alkyl chain or close a ring or do an atom swap. And then the craziest thing that they do is they call it scaffold hopping. So they're in there tweaking them all. Tweaking means round after round of synthesizing, testing, back and forth, chemists and biologists. But sometimes they sort of get between a rock and a hard place. And then they do this thing called scaffold hopping, where to me, it looks like a different starting place. But in their mind, oh, no, that's just by like reconnecting everything backwards, you know, and then they're back in the game. That's the real hard part of drug discovery, you know, is doing that. And, you know, it probably will come sooner in antibody engineering. But there too, it's very tweaky when it gets down to what makes a therapeutic clinical candidate, not just something that binds. So it's coming, but not this decade, probably not the next, where it just like takes over.
We shall see.
We shall see.
Let's thank Jerry for
Thank you all. Thanks online, folks.